A small yellow wildflower grows in the cracks of abandoned roads, on hillsides, in meadows across 5 continents. In 1996, a meta-analysis published in the British Medical Journal reviewed 3 randomized clinical trials and concluded that Hypericum perforatum outperformed placebo in treating mild to moderate depression. Commission E, the equivalent of the FDA for herbal medicine, formally approved it as a licensed treatment decades ago. The United States classified it as a dietary supplement, not a drug, despite the same body of evidence. 2 regulatory systems, 1 plant, opposite conclusions. This is Hypericum perforatum, St. John's wort. It has been documented in European medicine for over 2,000 years. It grows freely, costs nothing to cultivate, and resists drought. Its flowers bloom with mathematical precision around the summer solstice, every single year. The name itself is ancient. John's wort, from the feast of St. John the Baptist, June 24th. And yet, somewhere between medieval monasteries and modern pharmacies, its story became complicated. Later in this video, the answer to one question may change how history looks at an entire industry. No plant in Western botanical history carries a longer unbroken record of medicinal documentation than Hypericum perforatum. Hippocrates described it in the 4th century BCE. Dioscorides listed it in De Materia Medica, the foundational pharmacological text of the ancient world. Paracelsus, the Renaissance physician who shaped modern pharmacology, called it one of nature's most reliable nervines. Medieval monks cultivated it deliberately inside monastery gardens across France, Germany, and Britain. They were not guessing. They were preserving accumulated clinical observations spanning generations. By the 12th century, Hildegard von Bingen had documented its effect on what she described as melancholic disturbance of the spirit. The language was pre-scientific. The observation was not. This pattern, empirical use preceding chemical understanding by centuries, defines the story of St. John's wort. What monks called spiritual restoration, 20th century chemists eventually named hypericin and hyperforin. The compounds were always there. The vocabulary arrived later. History did not wait for laboratories to confirm what healers already knew. Hypericum perforatum produces a complex chemistry that modern pharmacology has spent decades attempting to fully decode. The primary active compound, hypericin, is a naphthodianthrone pigment responsible for the plant's characteristic red oil. Crush a fresh flower between 2 fingers. The deep crimson stain that appears is pure hypericin. A second compound, hyperforin, has been identified as the principal modulator of neurotransmitter reuptake in the brain. Peer-reviewed studies published in Pharmacopsychiatry established that hyperforin inhibits the reuptake of serotonin, dopamine, and norepinephrine simultaneously. Most synthetic antidepressants target 1 pathway. This plant targets 3. Beyond these 2 compounds, the plant contains flavonoids, xanthones, and essential oils that appear to act synergistically. Synergy is the word pharmaceutical chemistry struggles with most. Isolating one compound is straightforward. Replicating a whole plant matrix is not. The yellow flowers contain the highest concentration of active compounds at the moment of full bloom. Traditional harvesting calendars aligned perfectly with this peak window, not by accident, but by long observation. The plant essentially announces its own optimal harvest with color and scent. Nature does not require a laboratory schedule. In 1984, Germany established Commission E, a scientific body tasked with evaluating traditional herbal medicines using modern clinical standards. It was one of the most rigorous regulatory reviews of botanical medicine ever conducted. By 1984, over 300 clinical studies on Hypericum perforatum already existed in the European medical literature. Commission E reviewed them, applied strict criteria, and issued a formal monograph approving St. John's wort for the treatment of mild to moderate depression, anxiety, and nervous unrest. This was not alternative medicine. This was regulatory approval under a federal scientific authority. Germany's approval triggered a wave of clinical research across Europe throughout the 1990s. The 1996 British medical journal meta-analysis confirmed efficacy across 23 trials. A 2008 Cochrane review, considered the gold standard of evidence-based medicine, analyzed 29 trials involving over 5,000 patients. It concluded Hypericum perforatum was more effective than placebo and similarly effective to standard antidepressants for mild to moderate cases. The evidence was not marginal. It was substantial, replicated, and peer-reviewed. The regulatory divergence between Germany and the United States that followed was not a scientific disagreement. It was a jurisdictional one. In the United States, the Dietary Supplement Health and Education Act of 1994 created a category that fundamentally changed how botanical medicines were evaluated. Under DSHEA, a substance cannot be classified as a drug unless it undergoes the FDA's new drug application process, a procedure that costs, on average, between $1 and $2 billion. No one owns St. John's wort. It grows wild. It cannot be patented. Without patent protection, no pharmaceutical company has financial incentive to fund a billion-dollar approval process for a plant anyone can grow. The supplement category requires no proof of efficacy. Only a disclaimer. This statement has not been evaluated by the FDA. This is the precise sentence printed on most St. John's wort bottles sold in American pharmacies today. The same plant, the same compounds, the same clinical evidence. One label says, approved medicine. The other says, not evaluated. The difference is not chemistry. It is economics. This structural reality shapes which plants receive serious clinical investment and which ones remain in the supplement aisle indefinitely. The system does not evaluate based on evidence alone. It evaluates based on what evidence can be monetized. Hypericum perforatum is not a garden rarity. It is a cosmopolitan weed. It colonizes disturbed soil, roadsides, meadows, railway embankments, and abandoned fields across Europe, Asia, North Africa, and both American continents. It was introduced to North America with European settlers and spread so aggressively it was eventually classified as an invasive species in several Western states. The plant that 2,000 years of healers considered precious enough to cultivate deliberately now grows uninvited in places humans consider undesirable. The irony is precise. Agriculturally, it is a perennial. Once established, it returns every year without replanting. It tolerates poor soil, drought, and moderate frost. A single plant produces hundreds of seeds per season. Dispersal requires no human intervention. The summer solstice bloom is not metaphor. It is botany. Hypericum perforatum consistently flowers between June 20th and July 10th across temperate zones, a window so reliable it became embedded in European cultural calendars. Monasteries built their harvest schedules around it. The plant's biology is a clock. It has been keeping the same time for millennia. There is one area where both European regulators and the FDA agree completely. Hypericum perforatum has documented pharmacological interactions with prescription medications. Research established that the plant induces CYP3A4, a liver enzyme responsible for metabolizing approximately half of all pharmaceutical drugs currently on the market. This means it accelerates the breakdown of certain medications, reducing their effective concentration in the bloodstream. Documented interactions include antiretroviral drugs used in HIV treatment, cyclosporine used in organ transplant patients, and certain anticoagulants. These are not theoretical warnings. They are clinically confirmed and medically significant. The interaction data actually demonstrates the plant's pharmacological potency more clearly than any marketing claim could. A substance that interacts with prescription drugs at the enzymatic level is not a passive supplement. It is pharmacologically active. This biological reality sits at the center of every regulatory debate about St. John's wort. The plant is potent enough to alter drug metabolism, yet it remains classified as a supplement in the largest pharmaceutical market on Earth. The contradiction has never been formally resolved. It has simply been administered by label warning. The asterisk does more regulatory work than the science. The history of Hypericum perforatum is not a story about a forgotten plant. It was never forgotten. It was reclassified. The monks who grew it, the physicians who prescribed it, and the regulators who approved it in one hemisphere all arrived at the same conclusion through different methods. The plant works. The evidence, by any reasonable standard of evaluation, supports that statement. What varies is not the science. What varies is the economic framework surrounding the science. A wildflower that grows on roadsides and cannot be owned does not fit neatly into a system built around proprietary compounds. That is not a botanical problem. That is an institutional one. 2,000 years of continuous documentation represents a body of observational data that most modern clinical trials cannot match in duration. The summer solstice still arrives every year. The yellow flowers still open on schedule. The chemistry has not changed since Dioscorides described it. Only the context around it has. History suggests that what gets approved is not always what works best. Sometimes it is simply what can be owned.